Great. Well, thank you very much. And thank you everybody for taking time to sign in and listen to this webinar today. I hope it's a good discussion, especially at the end. And if there are any questions, I'm more than happy to address those in due course. So the focus of this seminar or webinar is going to be really looking at using the picture in front of you and thinking about how the vasculature reacts and responds to the tumor microenvironment. So hopefully in the image in front of you, you can see quite a large tumor mass in the background. And in the very front of this image, you'll be able to see the growth of a blood vessel that's starting to penetrate and feed into the deep, dark depths of that tumor microenvironment. And what's really interesting about these particular vessels is that they are relatively immature compared to the more mature pre-existing vasculature, which has a much more robust structure and a higher number of pericytes that give it more structure and obviously a better level of tone. These vessels that grow into the tumor microenvironment tend to be tortuous and leaky, actually acting to facilitate the metastasis and metastatic spread of tumors from this mass to other parts of the body. And so much of research that's being carried out now is aimed at looking at ways to stop the growth of these blood vessels, regress that vasculature, and hopefully limit both the growth of that tumor and its ability to disseminate throughout the rest of the body. And so just to give you a brief overview of what I'd like to talk about today, I'm going to give you a little bit of a summary about angiogenesis and the role of one of those growth factors involved in stimulating the growth of those more tortuous vessels, vascular endothelial growth factor, or VEGF. We'll discuss a little bit about the mechanisms of VEGF inhibition and, importantly, what the consequences are. So when VEGF is not able to signal and not able to fulfill its function, what are the cardiovascular consequences of that? And then really to get to perhaps the main point of this Synteca-sponsored webinar is to talk about two approaches that we certainly use in our lab. And we'll talk about pulse Doppler flowmetry and then some radiotelemetric studies that we've conducted, specifically looking at what are the cardiovascular responses to anti-VEGF therapies. So what happens when you block VEGF signaling? So just to give you a little bit of context about what angiogenesis is, both in health and disease, it's actually a critical process, and it involves the growth of these little vessel sprouts from a pre-existing vasculature such that we're able to feed into an avascular region. And it's vital in a number of normal physiological processes, so embryonic development, wound healing, and parts of the menstrual cycle. But what happens is that it becomes dysregulated in diseases, so particularly in the cancer environment, where that process is highly upregulated and is able to continue without inhibition. And so we end up with quite a substantial pro-angiogenic effect feeding into that tumor environment. So there's a big market for therapeutics that either target to promote VEGF and promote angiogenesis, or in the case of anti-tumor therapies to inhibit VEGF and cause a regression of that vascular structure. The receptor by which most of these angiogenic effects are mediated is VEGF Receptor 2, and we'll talk about that in a little bit more detail. So as I mentioned on the first slide, the vessel morphology of a pathological angiogenic vessel differs quite a lot to the normal vessels that we would see in a normal physiological state. So these vessels that tend to feed into this tumor microenvironment shown in the picture on the right are poorly designed, leaky, they lack vasoconstrictor tone, and the lumens are usually lined by a heterogeneous mixture of endothelial cells and tumor cells. And again, they don't have that adequate pericyte coverage, which helps with their lack of structure and the ability of them to be more penetrable by the tumor cells, which can then disseminate across to other regions of the body. So vascular endothelial growth factor, or VEGF, is arguably one of the most potent pro-angiogenic factors. It's a protein that's made by a number of different cell types. And interestingly, and I'll discuss this in a bit more detail later on, VEGF can be spliced into various isoforms. And there are actually two families of these proteins. One of them that functions, as I've described before, in a very much a pro-angiogenic context. And the other isoform family has the opposite effect in terms of promoting vascular growth and sprouting. VEGF itself has what I call the pro-cancer phenotype. So it promotes cell survival, proliferation, permeability, migration. And interestingly, the bottom point shown here is that it has been shown to have an effect in regulating blood flow and vascular tone. And I'm not sure that in the established male vasculature that particular point was really well researched prior to some of these anti-cancer therapies, anti-VEGF therapies in particular, coming to market. So as I mentioned before, vascular endothelial growth factor binds to VEGF receptor 2, and it tends to form a heterodimer at receptor 2, leading to a number of phosphorylation events at specific tyrosine residues within the intracellular environment. There are other VEGF receptors, and for those of you out there who are in the VEGF field, I'm not ignoring them entirely, but simply in the context of promoting angiogenesis, VEGF receptor 2 is probably the lead player. And just to look at some of the signaling pathways that are stimulated. So once these tyrosine residues shown here by these various numbers on the intracellular side of the receptor become phosphorylated, there are a number of downstream signaling pathways that are stimulated, resulting in some of those pro-cancer effects. For example, promoting migration, proliferation, and endothelial cell survival. And interestingly, there's an association between VEGF signaling pathways and nitric oxide release, leading to events such as an increase in vascular permeability and affecting vascular tone as well. So again, VEGF has a critical role, and we want to maintain these levels, particularly in the context of wound healing and the menstrual cycle. But when it goes wrong, then we have an issue that needs to be dealt with, and VEGF becomes pathologically upregulated in a number of diseases, and I'm going to focus on cancer here, but it's also involved in a number of retinopathies, autoimmune diseases, and as I said, cancer itself. So before I discuss it, there were a couple of members of the VEGF family. And hopefully on your screen, what you can see here is the signaling sequence for VEGF that is then translated into an active protein. In terms of exons being spliced in and being included in what would be later transcribed into and translated into the protein, there are 5 constitutive exons that are relevant to VEGF, including the VEGF binding region. So all 5 of these exons, 1 through 5, would be included in all of the VEGF isoforms. Depending on which VEGF isoform results, there is either preferential inclusion or exclusion of exons 6 and 7. And these really affect whether or not the released isoform is able to act more locally in the presence of exon 6 and 7, or is more diffusible and able to disseminate further away from the site of production. And what we've been really interested in is what is occurring with exon 8. And so the inclusion of exon 8A gives rise to what we term the pro-angiogenic isoform family, where the splicing event would include exon 8, and therefore you'd have a translated protein. Or alternatively, instead of exon 8 being included, you could potentially have a downstream signaling event that results, a splicing event that results in inclusion of exon 8B. And so when the proximal splice site is selected and you end up with VEGF of varying lengths, and I've annotated here with an A at the end, that is your typical conventional pro-angiogenic VEGF family. Or alternatively, we could have a splice variant that results in a very numbered of amino acids shown here, depending if exon 6A and 6B, 7A and 7B are included, resulting in a protein that has an altered 8' end. So if we have a proximal splice site selection, we end up with the pro-angiogenic isoform family. And if we have a distal splice site selection, we end up with the anti-angiogenic family of proteins. And I've just shown here that there are actually 6 amino acids that are resulting in either of those two isoform families that confer some structural changes to the protein and therefore affect how those various proteins might signal. So just to give you a little bit of data now and to describe some studies that we carried out. This was work that I did actually quite some time ago as a postdoc, working with Dave Bates down in Bristol. And we used a rabbit corneal angiogenesis eye pocket assay. And so on the right here, it's easier to describe it using the pictures. We took some MCF7 breast cancer cells and overexpressed either the pro-angiogenic isoform, so VEGF 165, which is arguably the most potent VEGF isoform, or we overexpressed a population including the anti-angiogenic isoform family, shown down here, annotated with the ending B. And we also had cells that were just transfected with an empty pcDNA3 vector. And so if you put in equal amounts of cells into a little slit made in the cornea up here, and then waited a little bit, over a course of 2 weeks nearly, what I can hope to convince you of is that you can see that there is a growth of blood vessels that are starting to migrate and move to where that slit was made. So where the pro-angiogenic isoforms were implanted into the rabbit eye, you can see that there is quite a robust pro-angiogenic response, even within the scleral tissue below. And in contrast, if you look at the picture in the bottom, where you make the same sort of slit, and in this case, in addition to the pro-angiogenic isoform, we have cells that are implanted in equal number with the anti-angiogenic, or potentially at the time, anti-angiogenic isoform family. Hopefully I can convince you that there was very little, if any, growth of those blood vessels towards that site. And so over a period of the 2 weeks, we can divide those areas up and afford them with an angiogenic score. And that's just shown here in the figure numerically. So where we have the empty pcDNA3 vector and the VEGF 165, the pro-angiogenic isoform, you can see quite a significant angiogenic response, and you don't see that at all in the presence of the anti-angiogenic isoform. So this was our first evidence that there was something happening where with different isoforms we see different cardiovascular effects. So at this point, I think, Martin, I'll hand over back to you if that's okay, and perhaps you could open it up for an audience poll.
Great. Thanks, Jeanette, very much for the first part of the presentation and for the introduction to the role of VEGF and consequences of VEGF inhibition. I'm certainly convinced, as per the last slide. So yeah, we'll give you a little bit of a break and run just another quick poll. We'd like to know, for those of you in the audience, which of the following animal models you use as part of your research. And again, you can select one or more of mice, rats, rabbits, or large animal models, be it porcine, canine, or non-human primates. So we'll just give Jeanette an opportunity to catch her breath, and then we'll continue on with the second half of the talk, which will focus more so on the cardiovascular consequences of anti-VEGF therapy. Okay. Very good. Okay, Jeanette, so I think we've closed our poll and we're ready for you to continue on with your talk. Thanks.
Great. So that was just really, as Martin very kindly said, to give you a little bit of context of why VEGF is important in terms of promoting or inhibiting the growth of those blood vessels into an avascular region. What I'd now like to do is talk about what are the consequences and what are the cardiovascular effects of some of those anti-VEGF therapies. So what happens when we block VEGF, particularly in the clinic? And this is really just a summary slide here showing that when we inhibit VEGF signaling via receptor 2, there are a number of consequences to that. There's an increased likelihood of capillary rarefaction. We start to inhibit some of those nitric oxide stimulating pathways, leading to vasoconstriction. And overall, certainly clinically, we end up seeing patients with an increased systemic vascular resistance and overall elevated mean arterial pressure, systolic pressures, and diastolic pressures. And so some of the issues related with anti-VEGF therapies are now quite well documented and include aspects such as cardiotoxicity. So again, relating back to that elevation in blood pressure. There's left ventricular dysfunction. And indeed, some of the anti-cancer therapies have a direct effect on the myocytes in the heart, leading to increased apoptotic pathway signaling and cell death of some of the cardiac tissue. And so cancer patients now have a significantly higher risk of potentially dying from some of the cardiovascular related issues rather than potentially the cancer itself. And so it's a major clinical issue and something that I think that as basic scientists we should be looking at to better understand what are some of the mechanisms that underpin these clinical conditions and clinical issues. And so just to expand on that a little bit, there are a number of processes that contribute to the development of hypertension and cardiovascular disease. And this slide, which I've borrowed from the publication shown below, I think really highlights some of these issues quite nicely. So with a reduction of VEGF receptor signaling, I've mentioned before some of the issues related to decreasing local vasodilator concentrations, issues relating to rarefaction, and then there are also issues relating to the kidney. And for those of you that ever look at the association between the kidney and the cardiovascular system, they're quite intimately intertwined. And so changes in kidney structure and function result in renal dysfunction. And again, these contribute to the development of hypertension and cardiovascular disease. So it's a major issue. And so as I was preparing for this webinar, I was asking myself, are these side effects actually surprising? And I guess to a certain extent, I came up with the answer that, yeah, they probably are. That under normal conditions, most of the endothelial cells are in a quiescent phase. And so the anticipation was that if we were to knock out VEGF signaling, that there wouldn't be any major cardiovascular consequences to that. But I do think that the clinical experience has perhaps changed some of these expectations. And the second question that I was considering is do these anti-VEGF therapies actually work? And I guess in answer to that, I would say yes, in part. But I think that the therapies are actually proving to be more challenging than originally expected or anticipated. So VEGF receptor tyrosine kinase inhibitors, when they're used on their own in cancer, they tend to not work particularly well. And so most of them are used as adjuvant therapies alongside a chemotherapy. And some patients are either refractory or they acquire resistance. So some patients just never respond to VEGF therapies in the first place. And many of those that do over a period of time lose that effectiveness and actually become more resistant to the treatment. And one of the major issues associated with that is that often, if that's the case, then the tumor becomes increasingly aggressive and invasive as a consequence. And so there really is a need to better understand and identify those individuals for whom these treatments are probably a better first choice and for whom it's perhaps a more personalized or targeted therapy. And so we undertook some preclinical studies in our lab using a rat model, really so that we could look at the regional hemodynamic consequences of blocking VEGF, either in an acute model or over a longer period of time. So I'm going to describe first the approach that we used using Doppler flowmetry. And in this case, we treated animals over a period of a maximum of 4 days. And then I'll move on and talk about some of our radiotelemetric approaches where we've been able to look over a much longer period of time, including an off-treatment period at the end where patients were no longer being actively treated with the anti-VEGF therapy. So just to describe the system that we use, it's actually quite a novel and bespoke system unique to Nottingham, which was originally developed by my PhD supervisors some time ago. And I guess I came back to inherit the family empire. But the system that we use, I've shown 3 examples here of different probes that can be implanted to obtain measurements of distribution of blood flow. And hopefully what I can convince you is that in the bottom example, which are the pulse Doppler probes that we use, originally these came from Crystal Biotech and now by Indus Industries. We use this small probe that's much lighter, so it's 0.2 grams compared to 2.8 grams of other commercially available probes to surgically implant these around 3 different blood vessels in our animal model. And so just to say that the Indus probes that we now use have a really nice silicon cuff. They're really lightweight. And so for us, it's really as well as being able to measure in multiple vascular beds simultaneously, it's also a nice surgical refinement because we don't need to make such a big insult and tease away so much tissue in our individual animals. So it's a really, really nice way of undertaking these studies. And so I'm very lucky. I have a fantastic senior research technician who actually helps us. And she in the past, she's constructed all of our probes almost from scratch. So we receive the crystal attached to the wires, and she actually makes our probes for us so that they sit at a 45-degree angle to the vessel. And just to show you and contextualize it a little bit, this is what one of our probes looks like, and that's the point of a ballpoint pen. So they're really small, they have a lovely soft silicon cuff, and we can surgically implant them around multiple vessels within one animal. And from this, we're able to use our Doppler flowmeter system to ultimately measure the Doppler shift. And so just looking at this equation, and I'm not a mathematician, so if you're going to ask loads of questions, maybe avoid this one if you're kind. But given that we're able, this single crystal acts as both an emitting and receiving crystal, we're able to measure the transmitted frequency, and then it's receiving it at a slightly shifted value. And if the velocity of the fluid and the sound in the fluid are the same, and the angle is held constant, because and I'll show you in a minute, but the probes go in, and then we don't start our studies until about nearly 2 weeks later. And during this time, a really nice fibrous cuff forms around the site of probe implantation, holding that probe at a nice angle. Then our Doppler shift value is able to give us an index of the blood flow velocity in the downstream vascular bed. And so just to give you a quick summary of our protocol, we implant 3 Doppler flow probes into a single rat. We wait roughly 10 days or up to 2 weeks. And then the animals undergo a second surgery where they're implanted with one arterial catheter and 3 venous catheters for drug administration. And the venous ones go into the jugular vein. my senior research technician actually creates these in-house and sews them up for us really nicely so that we have these lovely soft harnesses for the animals to sit in. They're attached to a swivel system so that we can keep our arterial lines patent. And then 24 hours after the catheterization surgery, we're able to start our monitoring. And so this is what our system looks like. On the right, we have the rat who would have a laparotomy and expose the vasculature into which we'll implant 3 different flow probes. And so we implant around the mesenteric, around the renal artery, and just at the descending abdominal aorta, hopefully as close to the bifurcation as we can, we put in what we call the hindquarters flow probe. And then these are attached up to our pulse Doppler system. And really, this is where we started our interaction with Syntica and with Indus, is that we asked them to develop for us this bespoke and novel 3-channel system so that we can simultaneously measure in one rat all 3 changes in vascular conductance through those different vascular beds. And so this new pulse Doppler box that we've received allows us to generate signals. And so we'll have the aortic signal, the mesenteric signal, and the renal signal. At the same time in this rat, they're also implanted with that arterial catheter into the vein, into the tail artery, which is hooked up to a fluid-filled pressure transducer, amplified, and that allows us at the same time as measuring those different Doppler shifts in the 3 vascular beds, we're able to measure changes in mean arterial pressure and heart rate. So we have a really nice picture of what the vasculature is doing and how it's responding. And by using these 3 different vascular beds, we're able to capture about 70% of the cardiac output from an individual animal. So just to give you a little bit of data, and I promise I won't overkill because I'm aware that I'm restricted by time, but we are able to make those measurements in a single animal and over a period of 4 days. And so we conducted some studies, and one of them was to look at a compound called cediranib. So this is a receptor tyrosine kinase that, amongst targeting other receptors and other signaling systems, also targets VEGF Receptor 2. Originally, it was developed as an adjuvant therapy for ovarian cancer. I think it's being repurposed at the moment. But one common clinical side effect, as I've mentioned earlier in my talk, was the development of hypertension in patients that were treated with cediranib. And so we carried out, so a PhD student of mine, Joe Carter, some time ago, carried out some in vitro experiments to look at whether or not cediranib was able to knock down VEGF responses. So she used an NFAT reporter gene assay, stimulated it with VEGF, shown along here, and looked at 3 different concentrations of cediranib. And hopefully I can convince you that she was able to knock down that VEGF-mediated response. We then moved on and conducted some in vivo studies, and they're shown here. And just to explain, because it's quite a complicated looking figure perhaps, along the top we have changes in heart rate and then mean arterial pressure. And then below are the changes in vascular conductance through the 3 different vascular beds that I mentioned on the previous slide. And so the dark colored circles are animals that are treated with vehicle, and the open circles represent the group, a single group of animals that were treated with cediranib. So we've got a vehicle and drug treatment for each of the different variables that we were following. And just to give you the protocol, the animals were treated with a bolus, an infusion dose of cediranib for an hour, the infusion on days 1, and then 24 hours, that's the beginning of day 2, day 3, and day 4. So this is over a 4-day time course. And hopefully what I can convince you is that by the start of day 2, we start to see a hypertensive effect in those animals that are treated with cediranib, and it's sustained over the duration of that short-term study. Not very much in terms of changes in heart rate. It's a little bit variable and a little bit all over the place, but perhaps a slight decrease. But significantly, there were vasoconstrictions that we observed in all 3 vascular beds. So if you see that it's starting to go more negative, that's indicative of a vasoconstriction. So we have vasoconstrictions in the renal, mesenteric, and hindquarters bed for those animals that were treated with the anti-VEGF therapy. So we then ran another study looking at a different anti-VEGF drug. So this is sorafenib or Nexavar, which is clinically available. Again, it's multi-targeted, as are most of the anti-VEGF therapies used for cancer treatment. And it's currently used in the treatment of advanced renal cell carcinoma and hepatocellular carcinoma. So Joe very kindly did the same in vitro style assay. Again, it's an NFAT reporter gene assay. And with increasing concentrations of sorafenib, hopefully, again, you're convinced that there was a dampening of that VEGF response. So we then moved into the in vivo setting and repeated the same study. So changes in heart rate at the top, the mean arterial pressure response shown in panel 2, and then the 3 vascular conductances below. And again, followed the same sort of protocol, treating for an hour on each of 4 consecutive days. And again, we noted a pressor response. So there's a hypertensive response that is developing over that 4-day period. But interestingly, perhaps less of an effect in the renal and mesenteric vasculatures, and a more sustained effect in terms of the hindquarters. And so we note that sorafenib caused a pressor effect and vasoconstriction, but that was most consistent in the hindquarters. It's still evident in the other vascular beds, but perhaps just not as consistently. And so we also did some studies where we tried various inhibitors or various antihypertensive therapies that we tried to treat at the same time. So this is just showing you a study that we did over a couple of days. And we, shown in the colored circles, we have cediranib being administered at the beginning of day 1 and the beginning of day 2 in the presence of vehicles. So that's the black circles. And the open circles is cediranib treatment on those days, but given at the same time as losartan. So losartan is an angiotensin receptor 1 antagonist. And what you can probably see here is that we weren't able to offset that rise in mean arterial pressure, and we weren't able to offset the changes that we were noticing, particularly in the mesenteric vascular bed. And so we tried this not only with losartan, but also inhibitors of endothelin-1 and other catecholamines, and we weren't able to inhibit particularly the pressor effects that resulted. And so I think there is a need for us to go back and really start to look at some of the mechanisms that underlie these effects and really unpick what's happening in perhaps particular vascular beds, and then to consider our anti-hypertensive approach to treating patients who are being prescribed anti-VEGF therapies. So just to move on quickly, we also conducted some longer term studies using radio telemetry. And I know many of the individuals that have joined the webinar today have probably had perhaps even more experience than I have using this approach. But just very quickly, animals are implanted with a radiotelemetric device that is able to monitor a number of variables, so temperature, movement and activity. You can put ECG leads in and get an ECG recording, and as well mean arterial pressure and heart rate. And so we used this system where the animals were implanted with one of these small transducers. They sit on individual cages, and in our case, they are all put with an uninstrumented animal as a buddy to keep them happy and calm. And we're able to monitor them over much longer periods of time. And so just to give you an example, and I won't go through all of these figures, so please, don't be upset that there are 6 images on screen. But for those of you that are interested in this type of study, we dosed animals with vandetanib daily for 3 weeks. And then we also followed them for a 10-day off-treatment period. And so if we just look at the top right-hand panel, we have a change in, we have the mean arterial pressure, and hopefully what you can see is that when treatment starts at time 0, there's a change in mean arterial pressure, so a pressor effect in those animals that are treated with vandetanib. So at the bottom, we have the vehicle animals, and at the top, we have those that are showing a hypertensive effect when they're treated with another anti-VEGF therapy, vandetanib. And what was quite interesting to us was that if you follow the change in mean arterial pressure into the off-treatment period, actually it was sustained over that 10-day period, and didn't overall, it didn't come back down to baseline. So whether there are some underlying structural changes that are happening or not, that's something that I think in future studies we'd like to go back and have a closer look at. And so just to give you another example, so this time, instead of looking at the mean arterial pressure, we looked at systolic and diastolic pressures. So these are shown here as change in systolic and change in diastolic. And this shows that persistent effect, I think, quite clearly, where you have, during this 21-day treatment period, a clear elevation in mean arterial pressure. And then as it starts to decline during that post-treatment or off-treatment period, again, we still see that there's a significant difference between those animals that were treated with vandetanib and those that weren't. And so we followed them for a fair number of hours into that period, and again, it didn't come back down to baseline. So the studies that we've done, we've been able to show both acutely and over a much longer period of time some of those cardiovascular effects of various anti-VEGF therapies. Moving forward, we need to start to understand the mechanisms that underlie some of those effects, particularly in terms of changes in regional blood flow and blood pressure. And as I mentioned before, to look at different ways of treating patients with antihypertensive regimes that both allow the anti-tumor effects of their therapies to have the same effect, but also diminish some of those cardiovascular consequences. And so, as I mentioned in the last slide, we would really like to explore some of the mechanisms that might be involved in perhaps that more persistent hypertensive effect that we observed, particularly as we progress through day 10 following cessation of treatment. So just briefly now to talk quite generally, I've been considering the sort of the advantages and disadvantages of both of the approaches that we use. The radiotelemetric approach has some clear positives or pros associated with it. We're able to continuously record changes in blood pressure and heart rate, temperature, etc., over quite long periods of time. The animals are housed with a buddy in pairs, and they're freely moving, so they're able to move around the cage without any limitation. The cons to that system is that it's quite expensive to set up, and we're not able to look at regional changes in blood flow. And so that's something else that perhaps at the moment limits the capabilities of what we're able to do. The system that we use, so for our acute studies, involves, as I mentioned before, the chronic implantation of those Doppler flow probes around bespoke vessels. And it doesn't have to be the 3 that I've mentioned. We've done previous studies where we've implanted around the celiac artery, for example. So there's scope for varied study. And the pros associated with this method is that we're able to take a continuous measurement of blood flow velocity. We're able to look at up to 3 probes in a single animal. And in addition to looking at changes in mean arterial pressure and heart rate, we're able to get a global view of what's happening and what's changing regionally in terms of vascular conductance. But associated with this method, there are also some cons that we need to consider. So for example, our animals are singly housed, as you see in this a little schematic on the right, and they're tethered. So they're hooked up to a springed swivel system, and through this metal spring run the catheter lines that we will then either administer drugs or monitor their blood pressure from. So they're singly housed and they're tethered, which gives them limited activity, and it also measures blood flow velocity and not overall blood volume. So that's another aspect for us to take into account. And then I just put this example in. So this has nothing to do with the current research that I presented in this webinar, but I think it shows the power of the model quite nicely. And so this is an example of an endogenous peptide being administered. So where this dotted line is here, that's the point at which IV administration of urotensin took place. And this is just an example from a single animal, and I think it highlights really nicely some of the aspects of our studies that we're able to explore in more detail than perhaps others are able to at the moment. And interestingly, what I want to point out first and foremost is that overall there's relatively no change in mean arterial pressure. So this white dotted line along the top doesn't really change. And so if we were using our radiotelemetric approach, we might look at that readout of mean arterial pressure and tick the box that there are no cardiovascular consequences to this endogenous peptide. But if we look more carefully in terms of regional distribution of blood flow, hopefully I can convince you that there's almost an immediate vasodilatation that happens in the mesenteric vascular bed, and perhaps a more sustained vasodilatation, slower onset vasodilatation that happens in the hindquarters, so this white line here. And at the same time, there's a slight renal vasoconstriction. So we have temporal changes, we have regionally selective changes, and associated with these overall effects in the vasculature, we can see a tachycardia or an increase in the heart rate response as well. And again, with nothing showing along in terms of overall mean arterial pressure. So the two compensatory mechanisms here in terms of heart rate and what's going on in the vasculature are seemingly largely offsetting one another, but there are significant events that are taking place. And so the methods that we use using the Doppler flowmetry model allow us to measure both the changes in blood pressure and heart rate, and for measuring regional distribution of blood flow. And it's a method for us to be able to test a number of pharmacological agents. So we can look at endogenous humoral systems. We can look at mechanisms that are involved in vasodilator and vasoconstrictor function, and really explore some of these novel peptides in a conscious, freely moving animal. And so if there's any take-home message that I could leave you with, it's that the integrated cardiovascular responses allow us to look regionally selectively, in a temporally distinct manner, and also allow us to sometimes look at events that are clearly opposing. And so in one vasculature, you might see a vasoconstriction, and in another, a vasodilatation. And that those changes are not always apparent if you only consider changes in mean arterial pressure and heart rate alone. And so on that note, I just want to thank most of the people that contribute to this work. So Sheila Gardner was my mentor and PhD advisor, and her and Terry Bennett were the two that really set up this system initially. And I have a great team, including Julie, who's my senior research technician, who really runs the operation for us in terms of in vivo work. And some of the work that was presented today was done by Sam Cooper and Joe Carter. And we have a number of individuals that have supported us, particularly within that fantastic hemodynamics team. But we also work really closely with our cell signaling colleagues, led by Steve Hill and Laura Kilpatrick, and our BSU team, who look after our animals. Without them, we're not able to do any of the work that we do. So at that point, I think, Martin, I'll hand back over to you, and happy to take any questions.
Excellent. Thanks so much, Jeanette. That was a really, really exciting talk, interesting talk. We've had quite a few questions come in. So, and we still have about 9-10 minutes to answer some of those. So let's get started. And thanks again for the really fantastic talk. Okay, so Jeanette, you've mentioned a couple times in the talk, you mentioned capillary rarefaction at one point, microvascular rarefaction. Can you expand a little bit about that? What exactly do you mean by that, and why would that lead to hypertension?
Right, so rarefaction is where there's overall a regression in that vasculature. And so there are a number of studies that have been conducted looking at how in the tumor environment, treating with VEGF therapies leads to a normalization of the vasculature and effectively a pruning off of less well-established vessels. And so there are advantages to that in terms of delivering chemotherapy to the tumor microenvironment. If you are able to prune off those leaky and tortuous vessels, the idea is that those that are left are likely to have a slightly more established and more robust structure with higher luminal pressures compared to the interstitial pressures. And so from that perspective, it's actually quite beneficial. But if you're starting to prune back microvessels and smaller vessels that actually help us distribute blood flow and end up with slightly higher total peripheral resistances, then the theory is that actually by targeting some of those vessels and damaging them by taking away VEGF signaling, you're actually putting the patient at more risk, because they're likely to have an increased total peripheral resistance, and that would contribute to the increase in their systolic and et cetera blood pressures.
Okay. Perfect. Thank you for that. And just a reminder to everyone, feel free to submit your questions at this point in time through our Q&A panel. Again, if we don't get to them all during the session, we'll be sure to answer them after the fact. Okay, Jeanette, what effect does anti-VEGF treatment have on apoptosis? And do these also affect endothelial cell survival and proliferation?
Yeah, so in terms of apoptosis, there are several studies that show that when you inhibit VEGF signaling, then there are issues relating to cell survival mechanisms, and so there's generally an increase in apoptosis. And as I hopefully described in my talk, some of these effects are direct in terms of affecting cell survival within the cardiomyocytes. And so we end up with patients that are suffering from cardiac cell loss, contributing to some of the dysfunction that I described. So it's VEGF is actually known now to be a survival factor and affect the survival of the endothelial cell lining within the vasculature, and also affect the responses of that microenvironment in terms of vasoconstrictor or vasodilator tone.
Okay. Excellent, thank you. So, Jeanette, and I think you can see the list of questions that have come in as well. Pierre actually has asked a couple of questions.
So yeah, there's an interesting one asking about the relationship between VEGF receptor 2 inhibition in terms of IC50s and systolic blood pressure. And that's a great question. We haven't done that. We haven't done that assessment yet, but I think that would be something that would be really important for us to look at. One of the problems that we've had, and in fact, I would welcome anybody that might have advice for our group. Actually, one of the problems that we've had is that the drugs that we've tested in vivo have been multi-targeted approaches to blocking tumor angiogenesis. So, for example, drugs like sorafenib don't just hit VEGF receptor 2. And so we don't have the specificity at the moment in our rat model. I know there are compounds such as I think it's DC-101, which is used in mice models, which is a more specific VEGF receptor 2 inhibitor. But what we're looking for actually is a more targeted VEGF receptor 2 blocker that works specifically at that receptor subtype, so that we could probably start to address some of those questions in the intact animal.
Okay, and I think there was a follow-up question to that initial question from Pierre that has just come in.
Yeah, so there's a question about have we tried Avastin?
Mm-hmm.
Or an anti. So yes. So one of the issues that we have with Avastin is that it's a humanized monoclonal antibody, and in the rat. So in my talk I described VEGF as the pro-angiogenic isoform VEGF 165 being the most potent of the members of the VEGF family, and that is absolutely true. But in the rats, VEGF is actually one amino acid shorter, so it's VEGF 164. And so to the best of my knowledge, Avastin doesn't work particularly well in the rat model, being a humanized monoclonal antibody. In terms of the anti-VEGF receptor 2 antibody, that's I think I probably started to address that in my previous question, in my previous answer, because the issue that we have is that some of these antibodies work really well in the mouse model, but translating it to the rat is quite difficult. And at the moment, we're not able to take what we do in the rat and translate that into the mouse. It's just it's too small for the work that we're trying to do. And mice move around a heck of a lot more. I mean, even our rats sometimes, try to climb out. So when we're not there yet in terms of translating into the mouse model. So I'm really curious if anybody else has tried something along those lines of a more direct or specific antibody or other approach. That would be great.
Okay, thanks for answering that question. And Pierre, hopefully that answers your questions. Just picking through the list, Jeanette, is there anything else here that looks, just keeping an eye on time, we have a couple more minutes. Is there anything else that could be answered in fairly short order? We can always leave some of these for after the fact as well. I guess I have a question for you. You mentioned, so you've placed those flow probes on the mesenteric, renal, and hindquarters, looking at the aortic beds, but why, perhaps a silly question, why those 3 vascular beds? I assume there was a particular reason for that.
Yes, well, in terms of what we're trying to capture are examples of regionally distinct vascular beds. And we're trying to capture as much of the cardiac output as we possibly can. And we also know that there is quite varied distribution of a number of receptor subclasses in those particular vascular beds. For this VEGF study, to be more specific, we know that there are issues relating to gut ulcers and changes in gut vascular flow in the human. And we also know that there are issues relating to changes within the kidney environment. And so those were 2 of the reasons that we selected those particular vascular beds, to see whether or not they were responding in a dynamic way to anti-VEGF therapies. And as well, it's always interesting to assess what's happening to the hindquarter. So you're capturing quite a lot of blood flow through that descending abdominal aorta, and to see whether or not there are significant changes where blood flow is either hugely increasing or dramatically decreasing, because that will have an overall effect on things like total peripheral resistance.
Okay. Very good. Well, we've reached the 60-minute mark on the dot. So I think with that said, And to be respectful of everyone's time, we'll wrap things up. And I just want to remind everyone, as we had stated earlier, that we have recorded the session and we'll make all the answers to any outstanding questions available after our talk today. So I'd like to thank Jeanette for her really amazing and informative presentation. Hopefully you all walk away satisfied that we've met your learning objectives. Jeanette, thank you really so much for participating in this. Some of the feedback we've gotten is extremely positive. We have a lot of our audience members thanking you and expressing that it was a great talk. So thank you for your assistance today and for sharing your research.
Well, and thank you everybody for your time.
Yeah, and so I'll mention that feel free to reach out to us. Our contact information is on the slide that's being presented if you have any questions about the methodology that was used today. You can also send any additional questions to us that we can forward along to Professor Woolard if you have any more questions for her. And we do also have some other solutions. I noticed through the polling that many of you are using tools like telemetry and echo ultrasound. These are all modalities that we also provide. So if interested, please visit our website and you can gain some more information there. So with that, thank you all for taking the time out of your busy schedules to attend our session today. We really appreciate it. And we look forward to seeing you at a future Syntac instrumentation event. Have a great day.